Okay. So let's look at our G protein. G protein coupled receptors are interesting. These are going to be interesting because they basically interact with both nicotinic and muscarinic. With G protein coupled receptors, we can get activation where we can turn it on, and we can get activation to turn a nerve off. Or we can get activation to cause an effector to turn on or an effector to turn off. And it's all based on those subunits that go into the G protein. Okay, remember we have alpha, beta, gamma, right? Some ligands, some receptors will be triggering the dissociation of the alpha subunit. And the alpha subunit travels through the plasma membrane and causes a specific event. Some things will bind with it, some ligands will bind to it, and it will cause the beta unit to disassociate and travel across and trigger a specific event. Okay. So with G protein coupled receptors, it's actually pretty fascinating here. So these are going to be more kind of muscarinic, all right, in that sense, where we have a binding with a specific neurotransmitter, a specific ligand. Okay. And we can either turn on or turn off our effector, turn on or off our nerve, turn on or off our muscle, depending upon the specific subunit that just breaks apart because of that specific neurotransmitter. All right. So, I mean, remember alpha, beta, gamma. And here we just have binding of one. Okay. We can have one. It can be alpha. It can be beta. Okay. If one disassociates, the other ones will run across. So depending upon which one breaks off, get you ultimately the various different actions that we see that we have with muscarinic. Because remember, 1 through 5, right? We had ones that turned on, and we had ones that turned things off, ones that stimulated and ones that inhibited. Okay. And the reason why, it depends on which subunit diffuses. Very short time these things act. Okay. Very short time frame in which these things are acting as well. Just like for all of them, to be honest. I don't know why they make a point to say that here. Okay. So there, acetylcholine binds to my receptor, my G protein coupled receptor. As you can kind of see, G protein travels across, binds to a specific ionic channel here, and in this case, you can see acetylcholine is, by the picture, shutting down the heart. Well, it's not shutting it down, but it's going to slow it down, right? And that's how that works in the heart. Specific ligand, acetylcholine, binds to a specific G protein coupled receptor, causes the G protein to disassociate and move across. There you can see beta goes across and stimulates a specific ionic channel. It hyperpolarizes. Now the heart doesn't stop, right? But it'll slow it down. And that's how the heart slows itself down. Okay. When you're resting and digesting. Body secretes acetylcholine. Those specific nerves going down to the sinoatrial node, the SA node. And that causes it to depolarize slower. It slows the heart rate, so it slows the heart down. Decreases the force of contraction by this G protein coupled receptor. Okay, and here I just have those words. Okay. So here in the heart, we had potassium channels opening up. Okay, again, it's that beta complex. It's that beta. But as soon as I see this opening up, what do we automatically think? Inhibiting, right? And if we think inhibiting, okay, that's an EPSP. That is a hyperpolarization event. And obviously the heart doesn't stop, right? We don't want that. But it just slow it down. If we look at the smooth muscle of the stomach, because remember, we are resting and digesting here, right? These are things that we do not control, involuntary, and that's what the autonomic nervous system is. In the smooth muscle, we close these suckers, and that's the alpha subunits doing. So it just depends on which one disassociates it. Is it alpha or is it beta? Well, if we close those channels, then there you go. That means depolarization. And that's going to be an excitatory postsynaptic potential. So it slows down the heart, turns on the stomach. And yeah, that makes sense. Okay. When you're sitting there eating, you don't want to be sitting there heavy breathing and your heart racing. Okay. If that's the case, if you're sitting there eating a sandwich and your heart's racing, you should probably go to the doctor, probably go to the hospital. You should not be happy. Right? So with these G protein channels, these G protein coupled receptors, okay, it depends on, again, the lock is different. Do we stimulate the beta unit? Do we stimulate the alpha subunit? If we stimulate that alpha subunit, we're shutting down those potassium channels. And that leads us to excitation. If we open up the channel, the potassium channel, obviously we hyperpolarize, so we inhibit it, make it more negative, make it more, create a greater, longer time to get back up to threshold so I can fire. And when it comes to the heart, slow it down. Okay, slow it down. Acetylcholine's everywhere. How do we get it out of that receptor? With acetylcholinesterase. Okay. It's pretty interesting how certain neurotransmitters can be removed when the presynaptic neuron is not stimulated. Sometimes that causes that presynaptic nerve to basically call acetylcholine home. So they'll re-up. As you can see from the picture there, acetylcholinesterase, it's ASE. Remember, whenever we see ASE, we always know it's an enzyme. Acetylcholinesterase breaks it down into acetyl and choline, or acetate, I guess I should say, and choline. That gets taken back up by the presynaptic neuron. And it just puts them back together. We recycle it. Some neurotransmitters get totally destroyed. But you can see from that highlighted picture there, we have acetylcholinesterase. It's just an enzyme there found in the surface of the plasma membrane. and it helps to break down acetylcholine. Because if acetylcholine is there in the synapse, what's it going to do? Bind to the receptor,
Bind to the receptor and be sent out.
trigger something to go. Okay. So this helps to break it down and destroy it. Break it down into its component parts, and it re-up it back into the synaptic knob of that presynaptic neuron. And we put it back together. And we recycle it. We do it all over again. EPSPs will have a slightly different name when we talk about acetylcholine with the somatic nervous system. Excitatory postsynaptic potentials in the somatic nervous system. We refer to these as EPPs, end plate potentials. Okay, it's the same thing. It's the same thing. Okay, it's just like an EPSP that we've been talking about within the nervous system here. Okay, from one nerve to the next nerve. Okay, it just happens to happen on a muscle. So we give it a slightly different name because that's what anatomy and physiologists do. You have to name everything differently. Okay, but the same jazz is happening here. All right, acetylcholine travels across the synapse, again, gets released from the synaptic knob. Okay, how did that happen? Again, as electricity traveled down the somatic motor neuron, it gets closer to the synaptic knob, triggers the calcium voltage gated channels to open, calcium rushes in. That causes those synaptic vesicles within the synaptic knob to start moving towards the plasma membrane of the synaptic knob. Causes the interaction of the V snares and T snares. Vesicle fuses with the plasma membrane and exocytoses the neurotransmitter acetylcholine. Diffuses across the synapse. Binds to receptors on the motor end plate. Okay. That causes a end plate potential. And remember, acetylcholine is always going to be excitatory here. Okay, it's nicotinic. So this EPP we now call just an end plate potential because it's happening at the motor end plate, only at the motor end plate of the sarcolemma. EPP does the same damn thing. Sodium channels open. Okay, sodium and potassium actually going through, but again, more sodium is entering, and that causes excitation, and that allows that action potential to now travel across the surface of the sarcolemma. Comes up to a T tubule, yada, yada, yada. You remember all that nonsense. But if we cause excitation at the end plate, if we cause an end plate potential at the motor end plate, we have excitation and the muscle contracts. So it's basically the same dang thing that we talked about with the nerve, from one nerve to the next. It's just now that we have a muscle. So we are compelled by law to give it a different name. And that EPP in the somatic nervous system is referred to as an EPP. Okay. Excitatory, or I'm sorry, end plate potential. Science is fun. I'm not going to ask you, but if you wanted to mess around with some nerve gas this weekend, there you can see what it does. It helps to inhibit acetylcholinesterase. So acetylcholine stays in there and it causes a constant firing of the nerve. So the spastic. Strychnine prevents inhibitory postsynaptic potentials in the spinal cord there. that inhibit contraction. So that's why you get, severe kind of contraction of everything. Don't get any movement. Botulism. Okay, if you're going to go get some Botox tomorrow, botulism, there you go, inhibits acetylcholine release, and that helps to stop the muscle from getting that wrinkledy, contracted appearance. If you're going to go out and shoot yourself with some curare, there it is. It prevents interaction of acetylcholine and nicotinic receptor. It, competes with it. So snake bites, if you're going to go out and walk around and find a snake in the snow, there you go. So fun stuff. There's tetrodotoxin there. Pufferfish. If you're going to go out for some sushi this weekend, that's why you dance with the devil with that one. Blocks voltage gated channels. Nothing will contract. Yeah. Okay. So just some fun stuff there. If you're going to play around with some drugs this weekend, there you go.
So EPSPs and EPPs are the same thing, just formed in different locations.
Yep. One's in the nervous system, EPSP, and one's in the somatic nervous system, which is in the periphery, and that's EPP. We do have a large menagerie of conditions here. The book goes into, I believe, several of them. Alzheimer's disease, it's associated with loss of cholinergic. I guess I should have just, this is a fancy way to say acetylcholine. Okay, it's just a fancy, it's an acetylcholine type of releasing cell. So it's associated with the loss of acetylcholine. And that's responsible for the degradation in memory because there's not enough acetylcholine being produced. And this happens, this process of lack of acetylcholine being produced and loss of those nerves, that happens a little bit later in the process. The loss of the acetylcholine neurons happens later in the process. There is a diminishing ability to form acetylcholine in Alzheimer's disease, and that can kind of happen somewhere, in the beginning a little bit, and in the middle it kind of snowballs, and then obviously at the end there, it's that and the destruction of those nerves. And you find this affecting really only the hippocampus region of the brain that's involved with memories. There's a few other areas involved with memory, and we'll talk about that next week probably. Myasthenia gravis. This one's an autoimmune condition. And the antibodies basically attack those nicotinic receptors. Okay, blocks them. And this is what can lead to some pretty significant weakness in facial muscles. Okay. And that's where you see it primarily being affected. There's some drugs that kind of help kind of bolster those synapses, kind of counteract some of the effects of myasthenia gravis. It's kind of interesting, but Alzheimer's disease and myasthenia gravis are just two of those different things that we affect, specific type of acetylcholine releasing neurons. So would I ask it on the exam? If I do, I'd ask it for bonus. Okay. I'd ask it as a bonus question. I do sometimes ask bonus questions. I don't label them as bonus. Okay. They're not, big bold letters. They may not say it. But I probably, when I'm typing up the exam, I'll put at the front of the question, this question is for bonus. Okay. And it requires mental gymnastics for me, but I sometimes do. And I like to put conditions as bonus type questions on the exam.